Vishu, please, Vishu's third lecture, where we will continue learning about stochastic processes.
Yeah, thanks. Is the mic working fine? Okay, excellent. Okay, so we will continue where we stopped last time. So just for a recap, this is not working. It's making noise. Should be closer to me. Is this better? Okay. So let me do a bit of recap and sort of set the broad goal for today. So today I have sort of two sorts of goals. One is to recap, summarize the general framework that I introduced last time on how do we go from stochastic microscopic interactions that are happening in biological world, how do we formally describe it using a mathematical framework. Okay, so that's the broad framework, but my goal really is not only the framework but to actually demonstrate the framework in some interesting examples. Okay, so now I realized that by the end of last lecture there were lots of very interesting questions and many confusions which are extremely common when you're reading and learning these stochastic processes and all these techniques for the first time. So I will spend some part of the let's say first half an hour giving some examples, clarifying those confusions. Okay, so that's the sort of first part of my talk. The second part, I want to explicitly sort of show, not via a whole lot of mathematics, because you can always go back, redo calculations and look at the papers for the detailed calculations, but actually show how stochasticity can change results of our expectations from deterministic models in some very interesting ways. Okay, so when I say stochasticity, for the purpose of today's lecture, I'm going to limit myself to stochasticity of small systems. Okay, so so that's the broad goal. Okay, so clarify a whole bunch of points about the general framework and then give you a lot of examples and including real data examples. Okay, that's the goal for today. So we start with so just to recap now. So every biological system has stochastic interactions. I don't think I need to convince any of you for that. So what we do is we try to write these stochastic microscopic interactions in some simple set of reasonably simple set of rules. And we did one such example in the last class, which was that of stochastic population birth and death on a lattice, right? We will revisit that lattice again today. And then, but moving to more general framework, once we have written down the stochastic interactions, we decide our state variable. Again, in the context of biological systems, these are often densities of various populations, right? And then once we have decided those state variables, we write down a transition matrix. How each of these stochastic microscopic rules are changing the state variable that is of interest. So here is the crucial important point. Now, microscopic rules are literally at the level of individuals. Two individuals interact, give birth, one of them dies, dispersal. So they are at the level of individuals. The state variables often are a coarse-grained variable. For example, I will not ask what happened to an individual, but I'm going to ask what happened to the something at the population level. What's the density of this species after this event, and that's what these transition rates capture. Okay, these transition rates are at the rate at a coarse-grained variable such as population density. Having done that, so master equation provides a very general framework. Okay, now realize that the processes are all stochastic, right? That means there are many possible outcomes. Like, if I toss a coin, head or tail is an outcome. Okay, if I toss the coin again, head or tail is going to be an outcome. So every outcome is stochastic. But we are now going to understand these possible events in terms of probabilities. Just like we ask what is the probability of getting 5 heads after 10 tosses, We now ask, what is the probability of population density being rho after some t time minutes? Okay, and that probability, as you can imagine, will keep changing with time because the births and deaths are happening all the time. Okay, master equation basically describes that. It basically describes, given transition rates between of our coarse grained variable, how does this probability density of our state variable changes with time? Now, this is a partial differential equation. This itself is not stochastic. Okay, because the way the probability density evolves actually is deterministic, although the underlying processes at the level of individuals are actually stochastic. Okay, so now the master equation is so general that it's often by itself not very helpful. So we need to, in some cases, if the transition rates are really simple, they are amenable to exact solutions. But when we have continuous densities, continuous times, often it's useful to make some approximations. And under some approximations in the context we were studying, such as, although technically when we started off with a finite population size, the density was a discrete variable under the assumptions such as, okay, assume a sufficiently large population, treat density as continuous, and then ignore some higher order changes and so on, we get something called a Fokker-Planck equation. Fokker-Planck equation, unlike this, is also, of course, this is also a partial differential equation that tells how the probability density changes over time. But the nice thing is it has only two terms. Okay, and this is manageable in terms of how we can solve. And today, unfortunately, I will not solve this equation. Okay, because again, more math, go back to papers, you will read them, you can read them about them. Now, here is the interesting thing. Okay, the Fokker-Planck equation has two terms. This term is called the drift term. The drift term is inspired by the idea of random walk. Whenever there is an imbalance in the probability of going to left or right, there is a drift, and that's precisely what the first term captures. And then there's a diffusion term. Diffusion term is what you expect from random walk. Things should just spread out over time, and this is a diffusion term. And this drift and diffusion terms, they're very interesting in the following ways. If you can sort of write the transition probabilities as some sort of a if you can sort of separate the transition probabilities as a transition rate that increases the density as t plus and something that reduces the density as t minus, the drift term is basically sort of an equivalent of birth minus death, whereas the diffusion term is, the drift is birth minus death, whereas the diffusion is birth plus death. So in some sense, the drift actually tells you what happens in an average sets, and diffusion tells you what happens around, what's the variation around your that expected average. Okay. Now again, as Priyanka was telling, a lot of this might be too much in a 5-minute summary. The point is, hopefully you can go back and look at this again and again and again and again, and you will get some insights about each of the term every time you look at this for sufficiently long time. Okay. Okay. So now, and once we have, now here's the thing, right? We started off with a stochastic microscopic interaction, and our interests are often in what is this state variable, how is it changing? But you know what? We have now made something abstract called a density function, and then we sort of moved away from the actual stochastic process in some ways. But we can actually go back to the full dynamics, the full stochastic dynamics of the state variable by going back to what is called an ETOSD, where you do describe how the state variable changes with time explicitly for each and every time you sort of execute this stochastic process. Okay. Now, again, all of this might be too much. So what I will do now is I'll just do a whole bunch of simulations. I have some simulations that are ready on my computer. I will do some simulations and sort of demonstrate some aspects of this. Okay. I will not solve these two equations explicitly. I will focus on this part, and I will focus on the last part. Okay. The solution of this I'm going to skip for today's lecture entirely. Okay, so let's do with an example. So before I go to example, any sort of quick questions, comments?
Question like, how is the ETO-SD, like, how is that helpful from the Fokker plan?
How is ETO-SD helpful? I will give an example, maybe it will be clear. Okay. Any other quick questions, comments? I'll try to answer them. Even if I don't answer now, I will answer them as I go forward. Okay. If there are no questions, let us start with the example 1, which is so all of you know what's a random walk, right? What is a random walk? Random walk is a very, very simple stochastic process where you have an agent who moves either to right or left with equal probability. So if the state variable is, let's say, x, of an individual at time t, okay, so how does what's the probability of now this can increase by a step length and decrease by a step length, right? So once whatever the step length could be some small values. So now again think of, there is no birth and death, this individual is not dying here, right? But think of birth and death in terms of the state variable. Okay, so if you sort of have x-axis like this, and moving to right will increase the x, moving to left will decrease the x. So in such a case, what would be t plus of x? What's the probability of incrementing x? It's half, right? In a classic random walk. What's that of decreasing? It will be half, right? So what will be f, the drift term? What will be diffusion? Diffusion will be 1. So what will be the ETO-SD? Basically delta x is equal to, so 1 over root n sort of captures, if you think of your space has some length and you have divided that into sort of n steps, I'm moving the small increment, it sort of is a measure of increment here. Okay, something about the variance, the step length here, this coefficient captures the step length. Okay, and then you have basically the g is 1, and what we do is, if I write this not in terms of if you write this in terms of this basically becomes the increments in the Wiener process. So with some, here the 1 over root n is less important. Let's call this sigma as the variance. Okay. So basically what you have is the ETO-SD basically becomes this. And what would be the Fokker-Planck equation? Fokker-Planck equation, the f will just go to 0. So the probability density would be some sigma square here in this case, because it's 1 over n times. So this is your well-known diffusion equation. Okay. So this tells you how each particle is moving. So the increment in the location is basically increment, this, as I already defined in the last class, this basically would be a Gaussian distribution with mean 0 and a unit variance. Okay. In fact, sorry, I have to be more careful. It actually is with the variance of dt. where dt is your increment, how much you are incrementing in time. Okay. And the corresponding equation that tells how the probability envelope is changing is given by the diffusion equation. Okay. So basically E to SD tells how the particles are moving. This tells how the probability density of particles is moving. Okay. So now let us do a simulation to understand this. So what I have done is I have done a simulation for you already. Okay, so what I'm going to show here is that initially the position of the particle is at origin. So there is a sort of a delta function at origin. Okay. And in this graph, what I'm going to show is how is this position changing over time. I will not show for a single trajectory, but I will sort of repeat this process. See, sometimes I might take right step and back to a origin. I might take 2 steps. So if every one of us executes a random walk, all of us would be moving slightly differently, right? So imagine that here I'm going to plot about 100 trajectories corresponding to 100 individuals. That's what I will do here. Here, what I will do is, how many of us are in various locations? You know, are most of us near 0 or most of us near 30? Where exactly are we located in this boundary? And how is this changing over time? Okay. Okay, let's do this. Okay, so I'll stop here. So these are about 100. I forgot now how many I was simulating, maybe 100 or 1000, I forgot. So you can see there are different, all of us are moving slightly differently. It's a superposition of all of us moving, all of our trajectories. What this shows is that most of us are actually near 0 after, oh, I forgot to plot the time here. Oh, time is around 50 to 60. So most of us are near 0, but some of us have gone far. Some of us have gone far to -20. Some of us have gone far to +20. And how far we are is actually increasing with time. And this is precisely what the Fokker-Planck equation does for you. Fokker-Planck equation tries to not tell you about the trajectories. So E to SDE tells you what the trajectories are going to be for each one of us. And the Fokker-Planck equation of the master equation basically tells how this probability envelope is changing over time. Is that clear to all of you now? And what is that solid line that I'm plotting? Can anybody guess?
Normal.
Huh? But where did I get the normal distribution from? From solving this. So I got the solid curve by solving this. I got the histogram from these runs of simulations. Okay. And you can see they're actually matching really beautifully well. And you know, the more the number of particles I use, better the match will be between the histogram and the probability envelope. Any questions, comments? So again, to clarify the relationship between E to SD and the Fokker-Planck equation, E to SDs actually talk about how each of the stochastic trajectories are changing over time. But every trajectory will be different. Now, if you look at the probability density of all the trajectories, that's what the Fokker-Planck equation helps you find out.
The relation between the solid line and the histogram.
Solid line is the actual analytical solution of this. Okay. Histogram is by simulating this for 100 or I'll tell you, I forgot, I have to look at my code. I think I'm simulating 1000 random walkers. So it's the histogram is based on the locations of 1000 random walkers. Any other questions?
The variance is your different one.
Exactly. Exactly. Yes. Where is the minus sign? Diffusion term doesn't have a minus sign. The drift term has a minus in the Fokker-Planck equation. Diffusion term doesn't. If a diffusion minus, it becomes a sink. Okay, so it's not a minus sign. Diffusion with the positive sign spreads. That's I mean 1,000 1,000 is a is not a really large number. Maybe there's a bug in my code. I don't know. That's also possible, huh? But it's a really good point, though. The point here is that the smaller sample size can induce biases. Okay, and in fact, part of the point that I'm going to make with the population models is that biases actually can, in some cases, be really important and actually can be very systematic. It's not just a deviation, but actually it's not just a case of wrong estimation, or it's not a case of higher variance. It actually can be fundamentally different. In fact, that's the point of the second part of what I want to convey you today. Yes.
Since you're just increasing time with a fixed number of walkers, that's why maybe you see more it not being very perfect to the Gaussian.
It should not be perfect to the Gaussian. In fact, it should not be. I would be really surprised and concerned if it was perfectly Gaussian with 1,000 particles. Like most physics simulations would use at least 10 power 6 or more, maybe. Yeah, I mean they do a good job these days. They're very good. So I wouldn't complain much. Yes. Any other questions? Okay. So that was the first example. Can I erase this now? Ideally, for the random walk example, I should just use the diffusion constant D when I'm using the analogy. I shouldn't use the N. So example 2 is the stochastic contact process, which is the stochastic birth death, which we discussed. So let's just recall what was the sort of, there was a lattice, right? And the lattice could have various zeros and ones. Okay. In fact, in the paper that I shared, which Akshit, my intern, must have shared with you all, the first paper from 2005, they don't do a lattice model at all. Okay. So, but I think there is a very specific reason why I chose a lattice model, to also demonstrate the idea of what is called a mean field approximation. Okay, because in real world, populations are spatial. We often write just ODEs and maybe SDs, like what I'm doing, and that necessarily requires something called a mean field approximation, meaning that any two individuals are equally likely to interact. Although it's not true, but we do it. I think this example makes that mean field approximation very, to me at least, very, very clear and evident. That was the reason why I chose this. So you could have zeros and ones randomly spaced to start off with, and then so the sort of the protocol for the stochastic update was I choose a site randomly, and if the site was 1, I choose a neighbor. I choose to do reproduction with the probability P if the randomly chosen neighbor is empty, but with probability 1 minus P, this focal individual dies. And again, just remember that nothing happens if you choose 0, nothing happens if you happen to choose 1, 1. Okay. So these are the two main rules. And then we also calculated these transition probabilities last time, and if my intuition is right, so the birth term is basically probability of choosing an occupied site, followed by probability of choosing reproduction, followed by the probability of choosing an unoccupied site as a neighbor under the mean field approximation. This requires a mean field approximation. I get the transition probability to be this, and likewise the depth doesn't depend on neighbors, it's just this. So there is no mean field approximation here, but there is an approximation here. So in the paper I circulated, which was one of my PhD students' paper, we have two variations. We have a death rate that is independent of P. Okay. And secondly, we also have an additional interaction, which we call positive feedback interaction. Basically, if you do choose two, the general assumption nothing happens in this model. So there we do two things. If there is a pair, one of the pairs is less likely to die, and that pair is more likely to reproduce in the nearby areas. So that sort of captures the fact that if there, for example, if there are two plants nearby, they sort of capture moisture better in drylands. So the seeds that fall near them have a higher probability of reproduction, of germination, and they themselves have a lower chance of death during droughts. That's called facilitation of the positive feedback process. So we also have an additional, more complex set of rules. For those of you who are interested in applying this formalism, go back, implement a more complex set of rules, and see if we can manage to understand all of this. Okay. Now with this, we know what is f of rho. What is the drift here now? The drift would be subtracting these two terms, right? And I think again, because I did this just 2 days ago, I sort of remember the terms. Rho over k, where k is 2p minus 1 over p, right? Okay. And then the g square of rho is, okay, it's actually adding these two terms now. Okay. So here is a very general interesting principle here. So both of them are stochastic, sort of, they are indicating something about stochasticity, right? So f determines how is your state variable drifting, right? So how is it drifting? It's birth minus death. That's the basic intuition behind drift being this. And what is the variation around it? The variation around it is basically the birth plus death. Okay, so variance adds up. So if you sort of want to connect back to your general understanding of the stochastic variables, if you take variance of two terms, it usually adds up, but the means just either add or subtract depending on the signs by which you do. Okay, so that's the sort of intuition if you want to build about what is drift and what is diffusion. Okay, so if you do this, I think if I'm right, we get this term. Is that right? So because I think what happens is p rho square term remains, the p rho and the minus p rho cancels, only the rho term remains. Okay. Yeah. So now if you write down the ETO-SD, how does it look like? It's d rho by dt is equal to, okay. So let me write it in the way that we often write, which is like a logistic model. I don't want to keep all the p's and stuff. So 1 over root n comes from this. So where r is equal to 2p minus 1 and k is equal to this. So the intrinsic growth rate connects to the probability of birth minus death. So p minus 1 minus p, that's 2p minus 1. and the carrying capacity connects to birth minus the divided by the probability of birth. Okay. Now here is the interesting thing in the model that Jacopo introduced yesterday with the demographic noise. What is demographic noise? It's a noise arising from stochasticity of the birth and death processes in finite populations. So n sort of represents the fact that our system size is finite. So n is the L by L in our lattice. It's a system size parameter. Okay. So larger the n, smaller the demographic stochasticity. And in addition, the demographic stochasticity depends on rho minus p rho square. He didn't have the term p rho square. And this sort of depends on the model assumptions. So for this model, you do have an additional term called p rho square. But you can also say that, rho square is probably sufficiently small because rho is probably small itself. So you can sort of ignore and then you get the demographic noise that Jacopo introduced yesterday. But here is the advantage of deriving from first principles. We also get terms that you did not expect, right? So this kind of a derivation where we start from microscopic interactions, systematically derive the sort of the coarse-grained equations. This is also sometimes called as derivation from first principles. Okay, because assumptions are very clear what we do, what assumptions I'm making, and I'm starting from very local level processes and then sort of scaling up. Okay. So is this now clear? So this is the etoSD, and of course the corresponding Fokker-Planck equation will be given by this, where you just substitute f and g square by the corresponding terms. Any questions so far? Okay, so now what I will now do is I will show you now.
Feedback. Do you effectively get like instead of a logistic model, sort of an alley effect?
Absolutely. If the positive feedback is above a threshold value, we actually get alley effect model.
Great, thanks.
Have you all heard of the alley effect model? Okay, yeah. So in fact, now the term here becomes now cubic. Okay. So now what we will now I will now show you two things. First, I will show you. See, now there are actually two stochastic processes here, right? One is the exact microscopic stochastic process I started off with, which is lattice based, and then there's an approximation at the density level that ignores the spatial structure. Okay, so I will show you simulations of both. Okay, so now one of my former students named Chandan, so he developed a website. You should actually go to this website whenever you have time. He has actually, out of his interest, whenever he was bored of doing classes and so on and so forth, he would pick up some model and do a visualization of it. Okay. So he has everything from physics, icing model in physics, to nuclear fusion, to complex system, firefly synchronization, collective behavior, Bernoulli percolation, Hilbert curves, whatnot. Okay. So now what I am interested in is this thing called contact process, which I introduced to you. So this here, there are, he has given us two control parameters. I can control the grid size. Okay, in his simulations, I can control the probability of birth. So this does not include this is the basic model. It does not include the positive feedback. Okay. And then, can you all see this better? Should I? Oh, me increasing the brightness here is not going to affect anything there. Okay. Okay. So, so the way the simulation works is I have to choose these parameters. What is the probability of birth? Let's say 0.3. Choose the grid size. If I choose a very large grid size, it takes a lot of time to simulate because it's a large space. So I will choose an intermediate size like 10 by 10. But when you do want to publish research papers, anything less than 250 by 250, people will throw it to the trash bin. Okay. So now what happens is if I click somewhere, I'm sort of germinating there. It's all empty right now. If I click somewhere, I'm germinating. And the red means that guy died. Okay. So P probability of birth is very low. So wherever I click, I am dying. So you remember the mean field prediction, the deterministic or the large. So I covered this in the last class as well, right? What's the prediction of the, if I ignore this, what is the prediction? The prediction was that the rho, which is the proportion of the cells covered as a function of P, for P less than half, it was 0, and it would reach 1 when P was 1. So this was the prediction of the model, right? Deterministic limit as a function of P. So clearly the model predicts that the approximation predicts that if the probability of birth is less than half, things are going to die. So that seems to be happening wherever I click. Sometimes there was a nucleation nearby, germination nearby, but it also died. Okay, now let's increase the probability of birth to 0.75. So you see what happened? So there I seeded one place, it led to dispersal. There are some deaths happening which are all the red flashes. The blue flashes are local dispersal and birth. And the gray, nothing is happening to them. So basically these are all updates. Basically there are two update rules, right? Whenever this happens, you see a red flash. Whenever this happens, you see a blue flash. Oh, the other way, blue and red, gray or whatever they are. Yeah, they're there. Yeah, because at any point of time, only one update is happening. Unfortunately, it doesn't plot rho as a function of time. I was hoping it would, and I was actually trying to now code this in the only language I am now comfortable, which is R. I used to do a lot of C programming once upon a time, and those days are gone. Okay, but it's very easy to make mistakes. I couldn't get that to work last night. It was already 1 a.m. and I said, okay, fine. Okay, so ideally, here is the thing, right? So we have a full stochastic model. For this model, I could also while doing this, I can also plot rho as a function of time. Right? I could have done this. It would have looked like this for a large rho. It would have looked like this for a small rho. For large P and small p. Okay. Okay, so this is the full model. Now the question is, these are all now approximations. Fokker-Planck is an approximation of how the probability density of this density changes with time, and the ETO-SD is an approximation of how the rho changes with time. So what I have done also, in addition to showing you this, is that I have done a simulation of this ETO-SD. Okay, and then I will now, just like for the random walk, I showed you a whole bunch of trajectories. I will now show you a whole bunch of trajectories. I will also again plot the histogram and see, let us look at the evolution of the histogram as well. For that, oh, I should have actually shown you the OU process, which I didn't. It's fine. We also did the OU process, right? Okay. So now I have set all the parameters, I think, correct? So I have chosen 0.6, huh? Let me choose 0.5. Okay, 0.55, which is just above the mean field model prediction. Okay. And I'm going to start with an initial density of 0.25. That means 25% of the cells will be occupied. Okay. And then run. So I have to wait a bit. Because the problem is the animation part takes a bit longer. It's not, the actual simulation doesn't really take long. Something happened? Okay. See, I'm starting with, so initial density was this. It was supposed to be 0.25. What happened? 0.25, right? Oh, I made a wrong name. It shouldn't be a OE. It's actually not a OE process. So I took the OE process and modified. So in the process, I think I forgot to relabel things.
Line 48.
Okay. I hope I have not opened anything. I'll just close these GIFs just to be on. And then I will also change the title if there is any that says, see here I am saying logistic model with demographic stochasticity. Okay, we'll just wait for a bit now. So here I think one file should come that's called demo logistic. I don't know why it's still okay. See, there are some strange. See, I think okay, the line is drawn wrongly. Oh, line is not drawn wrongly. Okay, okay, you will know what the line is. You will know what the line is. So the blue line is where things. So basically, at time is equal to 0. Initially 0.25 was the density. Okay, so now we have time 1 to time 2. One step has happened. So there are many trajectories which are now slightly diverging from. So I'm now again simulating about 500 to 1000 trajectories. Okay, with the same initial condition. Time 2. So they are again spreading. Different trajectories are slightly moving away. Let's now see where they move. So what's happening? Now, do you know what that horizontal? I for myself forgot what the horizontal line was. Do you now know what the horizontal line is? It's the predicted value of the, predicted value of the density based on the mean field model. But here is the thing. I haven't run this for long enough time, so I can't be really sure if this is correct. Okay. So now let's run. Actually, this actually should be correct. So I just haven't run long enough. Maybe we'll run this a bit longer, like 250 time steps, and I will also change to maybe 0.75 to make it a bit more interesting. Okay. Again, let's wait. Sorry. What P is the birth rate? P. 0.75 is what I have chosen now. Okay. Is it done? You can see where? Oh, here. It's still the old one or the new one? Modify 1216. Okay. Look, so I'm starting at again 0.25. So 0.75 is the predicted population. So you can see how both the trajectory as well as the PDF moved from the initial location. Now do you understand what part of this framework solves for which problem, right? So the Fokker-Planck equation tells how this density, okay, there's a density here, no? Histogram, how that histogram should move, whereas this tells you this is given by the ETO-SD. But here is the catch. Both of them can be wrong because coming from there to here required a bunch of assumptions. They're wrong. They could be wrong. They could be approximate. They need not be perfectly right. Okay, they could be sort of sometimes wrong. For example, as I told you the other day, if I do the full spatial simulation for p less than 0.6, everything dies off here. Whereas in the ETO-SD, it will survive. It will survive for anything more than 0.50. So it gets the critical point where the population survives wrong. But the overall characteristics, it does a good job. Okay. So here, unfortunately, I don't have that, the black line that was an envelope. that should have been a solution, analytical solution of this. I didn't have time to solve that, and I don't know if it even is. There's a closed form solution for that. Yes.
So in physics, occasionally even the existence of a critical point is sometimes
Exactly. Oh, okay, that's a good point. Okay, yeah. See, because the mean field approximation doesn't care about the dimensionality at all, right? So it turns out that in this case, for the 2D, there is indeed a critical point, and that is at p is equal to 0.61 and 25, something like that. Yeah, or 6025. Yeah. Yes.
Other than simulating the ICTS model, are there any other methods to see like how far are we from the critical point?
I mean, how accurate this prediction is?
Yeah.
You do better approximations. There are better approximations. For example, in the paper I circulated, there's something called a pair approximation. So mean field approximation assumes that any two lattice sites are independent of one another. It doesn't matter how close they are. Whereas in the pair approximation, you account for the possibility of who is your neighbor. Okay, because that does influence. For example, if there is a site which is 0, which doesn't have any ones around it, it cannot have a birth at all, right? Whereas the mean field model over predicts, doesn't matter where anybody is, there will be a probability of birth is equal to density. That's why it underestimates the critical point. Okay, it sort of over predicts the possibility of survival. Okay, yeah. So I hope now it's clear what's the difference between the different elements in this framework. Okay. So now having done this, okay, now I hope the first part of my talk is now over, where I wanted to convey you that how this framework works, what are the different ways to think about this, and what are the different parts of this approximations are doing and how they're helpful. Okay. So now the next part of what I want to do is why is this noise interesting? What are the interesting things that this noise throws up when we understand?
One more question, sorry. So the approximation comes into place when we derive Fokker-Planck equation, right? So if there
and then assuming in computing the transition rates.
If there are any ways to solve the master equation directly,
then there is no need for
it would be accurate.
Absolutely. Right, you're right. Yes. Okay. Okay, so now I'll try to I will stick to the same example as a starting point, and the next goal of my lecture today is to convince you why we should care about these types of stochasticity. And to do this, the first thing I will just state the result. I will not be able to prove this to you. Okay. So now when we wrote down the mean field approximation, which entirely ignores both the space and noise, what did we predict? So we made a prediction that as a function of the probability of birth, the density in the population remains 0 up to PC, and then there is a phase transition. This is called a phase transition or a bifurcation. Okay, and beyond P, the density increases continuously as a function of P. Okay, so this assumes it has ignored the noise entirely, right? Now, if we actually account for the noise term, which was there in the expression which I just erased, right? And then if you go back and solve the Fokker-Planck equation for stationary states. Okay, now I will not explain much more about what exactly it is, but you will find a very interesting result. It turns out that if this picture is true only if the population system size is more than some critical population size. Okay, so we know that this, we assumed n is equal to infinity when we derived this. We know that is true. But what if n was finite? It turns out that we still have this qualitatively correct even for finite n, as long as n is above some critical value. I will not bother what the critical value is, but what is interesting is that if n actually is less than some critical value, this nature of bifurcation actually changes. What you will find instead is something like this. Okay, don't worry whether who is above, who is below here. That's not the key point. But do you see the main difference between the two bifurcation diagrams? So what is the black one? What's the name for this? Huh? Okay, there are, if you use the terminology of phase transitions, you would call this a continuous transition. If you use the language of bifurcations, you would call this a transcritical bifurcation. Did you study about transcritical bifurcation in the last week? I don't know.
First lecture, so by now, yeah.
Yeah, probably too much back in time. Transcritical bifurcation, sorry, the black one, okay. The blue one, what do you call that? Fold or saddle bifurcation, right? In terms of phase transitions, you call them discontinuous transitions. So what this means is that accounting for stochasticity is really important, and our expectations of how systems will behave with the drivers can be dramatically different. So imagine if you are a a manager who is trying to understand an ecosystem. If this was the true mathematical model, you would think that change a little bit of, for example, birth rates and germination rates are dependent on things like rainfall, right? Other factors. According to the the continuous transition model, changing them would only change the vegetation cover in a gradual way. On the other hand, if you think of, if this is true, if this will be true in small populations, many real world systems are of small populations, right? And in fact, that's probably the norm. How many species have more than 1,000 individuals, right? Many species, and even if the global population is large, the local populations can be sometimes really small. In such a case, what this tells you is that there is some critical value of the birth rate below which the system can just collapse. The populations can just collapse, right? So the implications are really huge, depending on what model, what conceptual framework you're using to think about a real biological system, right? Okay, now this is of course a mathematical result, but with a very interesting implication. Okay, and if you want more details, the paper that I have sent has the entire derivation for this. What does NC depend on? What do you think it could depend on?
I mean,
there's only one parameter in the model.
There does only P.
There's literally only one parameter in the model apart from NC, like P. So I mean, and the P is the driver. So NC is an intrinsic parameter, just as P is in this case. It's a good question. Good question. I don't have a good answer right now because there are only literally two parameter values. I actually don't have a good answer. Yeah. Yes, absolutely. Absolutely, yes. Which was not the case in this. Okay, so this is one example of an interesting implication. So now I think I have I did try my own code just to demonstrate that I'm a very sincere teacher, but I took the help of AI tools, but even they crashed. By the way, just to be I learned something very interesting while I was using AI tools to generate the codes. Sometimes it can do flawless codes, but sometimes the same question, the output can be dramatically different, like literally like the stochastic process we're working on. And unless you know the answer, it would be very hard to debug. Because I have done this like a million times in my life, I know there was a mistake immediately when I saw simulations. But if you're a first-time user, you have to be extraordinarily cautious in using these without knowing what to expect. So I realized that there was two issues. In the simulation of the ETO-SD, it was making a mistake. Okay, there's a very subtle thing you have to account for when you're doing the simulation. That I immediately fixed. But it was using a package for animation that was much hard to debug because the package itself had a bug. So now how on the earth am I going to debug that, right? So that took a really long time to debug. So I was doing some really strange things in just showing the data was not really there. It was showing something in a sort of a periodic sort of way, in a very funny way. So I realized that took a lot more time to debug. Okay, but the first one would have been actually harder to. Both of them were in subtle ways hard to debug because you can debug the first one only if you know the algorithm and understand how it works. Second one you can debug only if you know what the results sort of should look like. So be very, very cautious when you're using all these tools. I'm using it as my own learning of how to use these tools. Okay. Now what I will now do is I will show you couple of more models and some data. Okay, now I'm going to show you a few models which are not the birth death models of the type we have described so far. So these are actually this sort of demonstrate the fact that I can use this framework in a variety of contexts. The key thing to think about are the state variables and how state variables are changing. Okay, so I'm going to show you two types of behavioral models. Okay, and then I'll then show you how we can actually use real data sets and sort of infer the importance of noise. And if I still have time, I'll show you one very recent work where we again show that the noise that arises because the system size is small can not only have implications for population dynamics like this, but also for evolutionary dynamics. Okay, in fact, it just changes what you think should happen. Noise will entirely reverse the predictions of our standard expectations of natural selection. Okay, so I'll show you some of these. I will not go into a lot of mathematical details. So this is sort of really more of an overview. Okay, so here is what is called a pairwise interaction model. Okay, so what happens in this model is that imagine there are lots of fish which are moving in some directions. So the micro states for each fish is the angle that they are currently moving. So for example, in this room, my micro state would be whatever this direction, let's call this north, whereas all of you will be towards south. So this is a nice system because I can approximate this system as a binary state model. I am going north, all of you are going south, looking south. Okay, but sometimes you feel a bit tired, you turn around a bit. Okay, so now likewise, now of course in this specific model the angle is continuous. Okay, so which makes it slightly more complex. That means the state variable for micro state variable is now continuous. So each of them have some heading angle, and then at some rate c, one of each focal fish copies a randomly chosen neighbor. Okay, so at some rates you can see what happened here. So this individual has turned towards the other individual. Okay, secondly, each fish or whatever that organism is can also change the orientation entirely stochastically, not looking at anyone, by some angle, small angle. Okay, we also have one more rule, which is sometimes when fish change the direction, they don't look at one random individual, they actually choose two random individuals to change the direction. And whenever this sort of these are called dyadic interactions, these are called triadic interactions. So whenever this triadic interaction happens, can you see what happens here? Who among the three is most misaligned? This one. So usually the misaligned one turn towards the other guys. Okay, so now the exact way you implement these rules are not very important. Okay, so another way to think about this is, again, in this example of, imagine just two directions. If three of us now are interacting, I am now more likely to turn this way than either of those two guys. Okay. So that's a question that we are not addressing here. We are just trying to understand here, why do groups synchronize their movement? So what are the behavioral rules they follow when they are synchronizing their movement? Okay, that's a great question. Any questions on the rules? Who is the third fish? So the thing is, as in the case of birth death model, at any instant in time, only one thing happens. In the next, so based on the rates determine how frequently events are happening. If the rate is very large, events are happening at a very frequent rate. The rates are low, they happen slowly. But at any given instant, only one can happen, nothing more than one can happen. So this is like a lot of, in this triad, it's like a majority rule. But as I said, you can sort of modify, as long as some version of majority rule is implemented, it's fine. For example, it could be average of these two. Okay, I might just align with one of the two. Both of them are sort of majority rules. So those details do not seem to matter a whole lot. Okay, you have a question?
Yeah, I'm trying to understand why you need the third rule at all, because
excellent question.
Yeah, I mean, the first rule already will make them align.
Excellent. So we will see that that's actually not always the case. So in fact, if you look at the classic models of, these are called flocking models. So there's a classic model of flocking dating to 80s by computer scientists and mathematicians, and the one that really popularized this was by a physicist named Thomas Wichek. So what they assumed in their models was that every individual looks around a neighborhood and does an average of their directions. So this is not averaging. This is a random choice of one individual. This is again not averaging. This is picking two in the neighborhood. What we show, what we will see in the later slides is that the mean field approximation of this model is same as the classic model. So basically anything more than two or more actually is same as averaging at the mean field level. So but it is not same as this. That's what I will show you now.
But you can drop one.
Oh, no, but these two are not same. So they are fundamentally different. They are fundamentally, yes, you do. They are fundamentally different. Yes. That's the whole point of what I'm going to present. Okay. Yes.
A small question. So in the first case, is there a notion of distance, or is it just?
There's no notion of distance in this model.
So that's the difference between these two. Here,
in both the cases, there is no notion of distance.
You said there was a neighborhood for the second.
So the neighborhood you assume is the entire group in both the models.
So there's no.
So there is no space here. So everybody can interact with everybody just for our analytical tractability. But if you do include neighborhood, Arshad has done work on that. That shows it doesn't really matter. Whatever I'm going to say will also be true even if all these interactions happened only with a small neighborhood. No, in this model, no. Okay. But you can choose, but the answer results will be still same under the mean field approximation, right? So it doesn't really matter for the mean field approximation. Somebody else, yes. Not two species, two individuals. It's all in. There's no space here, right? You can end here. You can think of it 10 dimensions. It doesn't really matter. But yeah, so in this model there is no space, but we have also implemented two-dimensional version of this. Whatever results I'm showing is perfectly valid even there. Okay, so let me go forward. So here I'm going to now divide into two parts. So I'm going to consider a case only the pairwise interaction is present. Okay, only this is present. I don't know why that slide came. I can't stop it. It's crazy. So don't look at this side. Okay, so look at only this side. Oh, what is happening? Some magic. Okay, as long as the slide doesn't go away, I'm okay. See. Okay, so it doesn't like me. So, okay, here, I'll just stop here. Okay. So, so if you now go through the same machinery, so we have defined the microscopic interactions in terms of how each individual is updating their state in terms of angles. You now define the transition rates in terms of angles, and then you write down a master equation, do some approximations, more algebra, much more complex, because the angle is actually a bit of a mess, much more messier to handle than the densities. Okay, so if you do go through the algebra and then derive an equation for, now I'm not deriving in, what's the equation I'm deriving for is very, very important. So I'm deriving an equation for a quantity called m that actually will be 1 if everybody is in the same direction. Okay. Is it clear? It's basically, think of it as vectorial average of all of our directions. Okay. So that's this quantity m. So I need to come from individual angles to individual vectors and then compute the average of all the individual orientation vectors, and then that's this quantity, and for that quantity this is the equation. Okay, now what does this equation tell us? Now I think so let's always whenever you have a stochastic differential equation, the first thing you should do is forget stochasticity. Okay, so which is, think of what happens without the stochasticity. So what happens without stochasticity? So remember, m is basically the synchronization parameter. If m is equal to 1, it means everybody is synchronized. M is equal to 0, they're all disordered. Everybody is in there. Because when everybody is in random direction, if I add all the vectors, what do I get? I get a 0 vector, right? So if there is no stochasticity, what happens? So the m is the synchronization parameter, basically will exponentially decrease, right? Now, of course, there is stochasticity. If there is stochasticity, what do you expect? Why would there be some alignment? Oh, don't see this. So what is your expectation if there is stochasticity? So at least my naive expectation would be that, of course, it decays to 0, but it will do in a stochastic way. That's what I would expect. But look at the equation here. This has an unusual form. In fact, it has a multiplicative noise. It has this form of, the strength of the noise is 1 minus m square. So if I were to plot the strength of this noise as a function of m, how would that look like? The strength of noise, basically g square, as a function of m. So one is synchronization. This is disorder. Okay. So if you look at this expression, when is this maximum? This is maximum when m is 0. And when is this minimum? When m is equal to 1. So basically, it looks like this. So the strength of the stochasticity is highest when the system is in disorder, and strength of stochasticity is least when it is synchronized. Okay. Now, if you remember, I showed one simulation in the first class. I know you have been hearing so many classes, so many things, and so on and so forth. Okay, where the analogy was that sort of somebody is rolling down the hill. Okay, but the stochasticity suggests actually is maximum here. What happened in this case? We actually found that the probability density function had a bimodality of this type, right? So I know you might have forgotten this, but the point is that so basically there is a tension between the stochastic term and the deterministic term. Deterministic term is trying to pull the system towards 0. But the stochastic term is highest here. So whenever stochastic term is highest, it just pushes away, right? It just doesn't care. But if it pushes so much, sometimes you can also end up being at a highly ordered state. And that's precisely, if you analyze this if you simulate this stochastic differential equation, whose code I will share you via Akshit, you will actually find that when the when the stochasticity is high, you can actually have synchronization, although deterministic model would have told you there is no synchronization. And just ask yourself, when is stochasticity is very high in this term? What is n? N is the group size. When n is small, when the group sizes are small, that's when you have high stochasticity. That actually means that you can actually have synchronization when the group size is small, but you will not have synchronization when the group size is very large.
Synchronization is transient or sustained?
It's sustained. In fact, it is sustained. In fact, what happens is that if you now again plot, let's now plot, in this case, let me do this mod m, okay? And I will do a probability density of mod m. What you will find is for large value of n, this is how it would look like. This is, let's say n is equal to 100 fish. If I now take 15 fish, what happens is that so if we actually take this stochastic differential equation and you write down the master equation and to solve the master equation for the stationary probability density, you will actually find this result. For large group size, you actually have results as expected by the deterministic limit. But for small group size, the results are not merely fluctuations, but they're actually dramatically different because this was disorder. So we went from a disordered state to an ordered state, which is not merely a small stochastic change, but actually is a fundamental qualitative change. Okay, so this type of schooling is called noise-induced schooling. Or noise-induced order in a very general way. Yes.
Why the first term of the synchronization is differentially because it looks like the copy beats the synchronization.
But the thing is, so what happens if you look at the mean field terms, the copying is symmetric, right? I copy you, you copy me, so it cancels out the overall effects. Because I could have been like this.
It's a little bit strange because if you imagine that 90% of your community is synchronized, and many more copying from 10%.
If you start there, what happens is that in the deterministic limit, so it turns out that if you write down the mean field model, so the pairwise copying, I copying you, you copying me, even when the group size, even if everybody is aligned, is unstable to the stochastic term which I mentioned in the previous case. It's like this term makes that me copying you, you copying him, he copying, all this basically, since we are just looking at pairwise, it is actually unstable to stochastic term. So in fact, the A term actually is equal to S. I sort of made a mistake here when I'm writing. Whereas, I just copied two different slides from two different papers. So with slightly different notations. So A actually is equal to S, the stochastic turning rate. So C is the copying rate. So copying rate comes only as a multiplicative noise term. And copying term tells that the stochasticity of copying is highest when you are.
Now it is.
And another way to think about this is, if all of us are indeed aligned, okay, then if I copy one of you randomly, I would still be aligned and all of you are still be aligned. So therefore, stochasticity of turning would be minimal. And that's precisely why when m is equal to 1, you have the minimum stochasticity. Okay. Yes. M is a vector in this case. Because average of the vector is a vector.
Oh, I'm so sorry.
Yeah. Okay. But you can also do a scalar version of it considering only discrete angles. And it's there in the paper for easy. So this is non-spatial model, right? So this is a non-spatial model. In the spatial model, yes. I do expect that. Can I just go forward? Yeah. So one quick question. Yes. You mean real world? Yeah, in real world, organisms are known to be in millions. So locust swarms can be in really, really large numbers. It's not applicable for those cases. So the model I'm presenting is more relevant for organisms which are also found in small groups. Okay. Yes.
you'll have a problem. And I'll explain you why. What initially confused me is the noise, which is in fact the copy.
Yes, noise actually.
Imagine we set up 8 to 0. You will still have a very, very noisy dynamics for M because it is multiplied by the noise.
Yeah.
However, my intuition tells me that if I synchronize the vast majority of positions, 95%, M will point to the direction of 95%.
Yeah.
For a long time. It will not change.
Yeah. You're right. So you are just changing the orientation. You will not change the synchrony. So the synchrony will remain in that case. So if you do set the A is equal to 0, the mod M will remain close to 1, but only the direction will change. Yeah.
So if everybody is synchronized and the noise.
Yeah. Okay. Yeah. So now the second on this side, I'm now in addition to this, I'm now incorporating the ternary interaction term. Let's call this rate as H, which is missing here. Sorry. You now see, you will actually now have an effect in the deterministic term only when this is present. You will now get HM times 1 minus M squared term. This is also for those who are familiar with. You will find this term even in the classic Wittek toner 2 approximations. Okay, so you will get this cubic term, which now will have deterministic order, even in the absence of stochasticity. But that is possible only when you have a 3-body interactions for alignment. Okay, so this is now, so take home message is that if you want to have schooling that is stable, with only pairwise copying, you do have to be only in small groups. Large groups, pairwise interactions are unstable. But the small group pairwise interactions can stabilize schooling entirely because of stochastic effects. But when you have 3-body interactions that induce positive feedback, even in large groups, you can actually have synchronization. So now, okay. So now, how do we now make use of this theory and apply this to data? So what I am now showing you on the side here are actually real data sets from the fish experiments we have done in our lab. Okay, so now, so we can actually do experiments in the lab where we can dump some fish in a fish tank. They will move. When they move, you sort of use high resolution videos, and from those you can calculate the same quantity, which is the average of all of their vectors, and you can now obtain a time series of this type. Okay, you can also plot the histogram. You will see that the histogram of the synchronization parameter is actually peaked at 1, meaning it's usually very, very highly ordered. So the question now to ask is this result happening because of 3-body interactions among fish, or is this because of the 2-body interactions at small group sizes? Which of them is the one that explains this data? So to really understand that, you have to go back to the tutorial that Akshit did, where he showed that if we have a time series data, even from very different models, but they look very similar in terms of the PDFs, even then we can actually distinguish the 2 time series data sets. Okay, so based on this, for example, here is an example. So here I'm showing you from the simulations, have done simulation of pairwise interaction model. Pairwise interaction model shows a bimodal distribution. So this is the synchrony parameter, both +1 and -1 means they're highly synchronized. So it shows high synchrony. This is the 3-body interaction model. Again, this model also can show high synchronization. And you can look at the time series, they look very similar. They look at the PDF, they look very similar, but can you really distinguish the 2? So the trick actually is entirely what Akshit showed you in the tutorial on Monday. We can use this package called PyDaddy, okay, and then basically can we infer such an SDE from the real data set? So far what we have been doing is I'm going this forward way, right? I'm going from the interactions, writing all this, and then come to SDE. But if I have only data, can I get the SDE? Okay, so that's what this package does. Okay, so which you have all now tried out, and I'm assuming you're all experts in this now. Okay, So now what we do is we apply this to real data sets. And when we apply this to real data set, we do find that the deterministic term is only linear, not cubic. Okay. And we find that the stochastic term is exactly of the same form that would be predicted by the pairwise interaction model. Okay. So this sort of strongly shows that in the real data, what we are finding is a pairwise interaction between fish, pairwise alignment interaction between fish, and the schooling is actually stabilized by the stochasticity of the small group sizes. And we would not be able to arrive at all these conclusions if I had entirely ignored the second term, which is stochastic term in the. So I want to emphasize this is a data-derived model. We had derived this model from microscopic interactions, but this equation in particular we actually got it from data. We didn't force fit this. Okay. So, and by the way, this is how the fish school really looks like. There are lots of fish which are moving with blue arrows on their heads. Okay. Have you seen fish with blue arrows? Okay. And then you can, based on those arrows, you can plot the average synchrony parameter. It's called the order parameter as a function of time. And from this time series data, we can actually derive the underlying stochastic differential equation. Okay. And that shows that the stochastic term is really, really crucial and important, and you cannot merely ignore that as, oh, it makes some noise around deterministic stable state. It actually is truly fundamental, and it changes the characteristic of the, of the system. Okay, so now done. I'll skip this. I have only 5 minutes. So if if you want to read more about these these are the papers that sort of explain all these. Okay. Finally, I have 5 minutes. I will just give you a very short glimpse. So moving from behaviors to populations to evolution. Okay. So I'm going to. This is a new paper we just published this month, where we looked at how does stochasticity from finite population affects evolution. The key message is there. It can change the direction of evolution. Okay, so I will not go through all the details. So feel free to go through if you're interested, but I will just show you one, again, the same formalism. There is a birth-death process. We write down the master equation. We derive the SDEs. Okay. Okay, so I'll not bore you with more math. Okay, cool. So any questions? So I will stop here. I will not explain how this interesting result happens.
Summarize the results again.
This result? Oh, this one. Okay, I can tell you. I think the next slide. I was wondering where is the damn slide of summary result. I couldn't find. I think. Here it is. Okay. So, have you heard of a replicator equation? Huh? What is replicator equation? It tells you how if there are different traits, trait A, B, C, D, E. Let's denote the fractions of them by pi, where I goes from A, B, C, D, E. The rate of change of this frequency is given by the fitness of the trait minus the average fitness of the population times the pi. Is that fine? So if the fitness of the trait is more than the average fitness, this is positive, therefore pi will increase. If it is negative, pi will reduce. So it will stabilize whenever the fitness of a trait becomes equal to average fitness. Now, is there stochasticity here? Should we include it or not? After I learned almost a second, we must include, right? Okay, so this is our version, which actually includes stochasticity. So now the first term is exactly same as this, which is what you expect. Okay, the last term is the stochastic term, Wiener process increment with a coefficient which is given by 1 over the population size. K times nk is the total population size. Okay, so the noise term is proportional to 1 over population size, which again makes sense. These two themselves are not super interesting, but there's something very interesting here which I'm going to ignore. Look at this term. We now have a new term in the drift as not the evolution drift, the physics drift. So we have a new term which adds on to the replicator equation, whose strength also depends on 1 over the population size. But look at this term. What is this tau? What is this new thing tau? So the rate of change of frequency should depend only on the fitness. Why does this depend on a new thing called tau? So what is tau? Tau is basically the summation of birth and death. We call this turnover rate. So fitness is what? Birth minus death, right? Fitness is basically birth minus death rates, whereas tau is the summation of birth and death. We call this turnover rate. Now, how do you understand this? Imagine, consider two species, two types. One has, let's say, high birth rate, high death rate. Okay, let's say difference is 1 unit. Let's say there's a second type which has a low birth rate, low death rate, but the difference is still 1 unit. So what will be the fitness of this and this? They will be both same. They will be both 1 unit. But the turnover rate, what about the turnover rate? The turnover rate of the first one where the birth rate was very high, death rate was very high is summation of the two. So tau I for the first type is more than the tau I for the second type. What this term tells you is that even when the fitnesses are same, this term will become 0. When the fitnesses are same between two types, there is this term which can change the frequencies. So the fitness, what do you expect? When the fitnesses are same, do you expect the frequencies to change? You don't expect, but not only can it can change in a predictable way. Meaning, in fact, there's a minus sign here. What this means is that the one with slower turnover rate actually undergoes a fixation. So that's the key result of this paper. Okay. Yes. Yeah, yeah. So I will just show you via a graph. It does matter. So I'll just show you via a graph. I mean, I don't know how it matters in the actual real populations, but I can certainly show you, in principle, how it can affect. So what is our intuition about evolution and drift, right? Let us say we have this is time. Let's consider just two types to make our life easy. P is equal to 1 here. Let's say I'm starting at half. Let's consider two populations which have equal fitness. What is our classic intuition about drift now? With 50% probability, type A will get fixated. With equal probability, the opposite can happen. What this equation tells you is the following. That is not true when tau 1 is not equal to tau 2. This is true only when tau 1 is equal to tau 2. So when the now let me now use two colors to indicate two different types. Let's now use black to mean lower turnover rate. So what it now shows is this is the, if tau 1 is less than tau 2, 1 will fix, even with drift term hanging around. Okay, so this is what is predominantly going to happen, not with equal probability anymore. Even if so, the one with lower turnover rate will actually undergo fixation, is what this predicts. Yes, and this is true only in small populations, very small populations. In fact, yeah.
So far in all the stochastic models with what you showed and what Jacopo showed, the turnover was coming as a multiplicative part of the noise, whereas here it's coming as a deterministic one. Is that because
I have hidden it is also there in the DWT term. If you look at the variance of this, so DWP is not the classic Wiener process. It doesn't have a unit variance. It does have a multiplicative noise here. So yeah, you're absolutely right. Even this will depend on tau. So here is the interesting thing. Why did it come inside is a very tricky, interesting question which we'll discuss outside. Okay. So you would have expected the multiplicative noise term to remain here only, but not in this part of the equation, right?
Is that because we're looking at frequencies? You shouldn't
expect the tau t plus t minus to be present here at all. But I will discuss this later. It's a very interesting point. Yes. So is the point clear? Basically, this is our classic expectation of drift under equal fitness. But what we are showing is that actually this can happen even if the fitnesses are equal in small populations. And what matters is this turnover rate. Okay. I mean, to be very clear, this is we are not the first one to show this. Okay. People have known this for a long time in various contexts. If you have heard of bet hedging, so one version of, so basically, to be very clear, this was known in the context of bet hedging. This was known in the context of some epidemiological models. What we have is a systematic first principle derivation that sort of very clearly delineates the effects using an Ito-SD framework. Yeah. Yes. Oh, we always use Ito. So that actually is a noise-induced drift. So I think, do I have that full term here? So again, just to be very clear, if the population size was very large, both the terms go away and we go back to replicator equation. Okay. Only when they're not present. So we call the noise-induced term as the indirect effect, indirect mechanism of noise-induced effects. This is because it averages to 0, but even that can have a non-trivial effect. This is what we call the direct mechanism. So this direct mechanism is what translates to classic bet hedging literature. Okay, where the variance of the fitness is sort of just put in without making connections to the turnover rates. Okay. Yeah, that's correct. Yes. Any other? Yes. Is there that's a good question. I think there could be. Yeah, because one is omega A B minus D, the other one is B plus D. There could be covariance. In fact, there will be. Yeah. But are there? Yeah, I mean, in fact, this was a question one of the reviewers actually asked. Maybe were you the reviewer? Yes. Absolutely. So can I go to the last slide then? This is my last slide. So noise is very cool and also ecologically important. Okay. So in fact, when I was doing my PhD, I was studying with my PhD advisor Jayaprakash, and he would always emphasize stochasticity can do really counterintuitive and interesting things. And I think right now, which is almost now 15 years, we are now finding empirical evidence to demonstrate this in an unambiguous way.